Hello, everyone. Should I start, Alma? Hi, Felipe.
Hi, everyone. So thank you, Lucas, for starting this great session of the PPCR Journal Club. This is session number 5, and we're really happy to have you here and to propose this great article that you're going to share today with our colleagues and PPCR alumni here. So Lucas, he was a student in 2019. He was one of the best students in his group and has been really active. Currently, he's a TA2 who's really engaged in the program, and we hope that you enjoy his section. So Lucas, you may begin.
Thank you, Alma. I would like to welcome all of you to the PPCR Journal Club. The topic today is going to be the HPTN 052 study. So welcome to the Journal Club. I am currently a medical student at FMUSP, and this year I am a TA2. As a disclaimer, I don't have any potential conflict of interest. And many of the pictures that are part of the slides are from Keith Haring, that was an artist back in the 80s and 90s. All right. If you have any questions, please feel free to raise your hands, or you can stop me as well. So let's start. This is the article that we are going to discuss today. When it was published in 2011, it was elected by the Science magazine as the breakthrough of the year. It was the first article published by this clinical trial, and many other publications came from this clinical trial. Awesome. So let's just first discuss the background of HIV and when the study was designed. So first, I'm gonna do a quick overview about HIV. HIV is a lentivirus from the family of retrovirus. It has a RNA genome, and it is the virus that infects human cells and causes AIDS. The origin of the HIV-1, that's the pandemic version, and the HIV-2 is another HIV virus that causes AIDS, but is more restricted in the African continent, is closely related to SIV. And it's believed nowadays that it originated from non-human primates in the early 2000th century. So to understand the origin of HIV and its introduction to the human populations, we need to travel back to early of the 20th century. The most accepted theory today is that it originated from a CIV. CIV are a family of virus that causes disease in monkeys. There are a lot of types of monkeys that have HIV. And it is believed that the HIV-1M was introduced around 1908 in populations on the Congo River Basin, mostly because of bushmeat trade hunting. So it's closely related with colonialism and with the expansion of the frontier between human habitation and the wildlife. So this crossover probably occurred around 1908, but the spread was dependent on the development of cities in the region and on sexual trade routes. So the virus from the West Africa traveled east to the East Africa in the 70s. And in a study conducted in 1986, already 85% of sexual workers in Nairobi were HIV positive. From East Africa, the virus traveled down to Sub-Saharan Africa, where most of the cases occurred. In the United States, it's believed nowadays that the virus came from the 70s, from somewhere between from Haiti. There was a lot of workers that traveled from Haiti to Congo, and then they went back to Haiti. And around the 70s, the virus was introduced in the US. Some landmarks of the HIV pandemic in the US are, so the first case reported by the CDC was in 1981. one case in San Francisco and another in Los Angeles. And there's a lot of critics about the prompt response of the US government. So today we are not going to have time to discuss about this early action of the American government. But there's this book called "And the Band Played It On" that discussed this very well. So I would recommend checking this book, and there is the documentary as well. In the United States, the HIV pandemic had a massive impact on communities like the gay community in Castro, San Francisco, or the villages in New York. The virus itself was discovered as a virus in 1983 in France. In 85, the first test was available, and in 87, AZT was approved as a treatment. When you're thinking about a disease, there's a tripod on controlling a disease. This tripod is about prevention, treatment, and cure. The most of the treatment, a better option of treatment was introduced in 1996 with HEART, and this was effective and life-changing. So we go to the reality when the study was proposed. This is in the early 2000s. So in the early 2000s, there were already good options of treatment available, and there was a huge discussion about when patients should start drug regimen. So there was this gap in the literature when would be beneficial to the individual to start HEART, and also there was a question if patients treated with HEART became less infectious. So people that had relationships with seropositive patients have a higher risk of getting HIV or not. So they proposed this study with the primary outcome of assessing if early treatment was beneficial in the prevention of HIV and serodiscordant couples. So the study design, it had two arms. One arm was composed of serodiscordant couples who received early treatment. So when they were enrolled to the study, they received HIV treatment. And the other arm received only when they had the CD4 count lower than 250, or if they had an AIDS condition. The primary outcome was to assess linked transmission of HIV between those couples. And the secondary outcome was to assess if there is a health benefit for the HIV positive patients. So as inclusion criteria, there were inclusion criteria for the couple. So they should be serodiscordant. The partner should be willing to disclose his or her HIV status. And for in the individual level, the HIV positive patient, he shouldn't have been receiving HIV treatment, and for the partner, he had to be negative for HIV. As this exclusion criteria, you can see that the couple, they could not use injection drugs in the last 5 years. They should not have been participating on a trial of an HIV vaccine. And there were another exclusion criteria as well, as you can check. So what is really different about this trial is that it proposed to test if the partner got HIV from his partner or not. This was a quite advance, and this brought many advances for the interpretation of the trial. But it was a really hard trial to conduct. It took several years, and for the participants, they had several visits during the trial. So at the start, they had monthly visits, and when they start treatment, they had treatments. They had to check their response to the treatment. So it was a quite hard study to conduct, but everything went well, actually. So for the assessment of seroconversion, they did something very nice, is that they sequenced the gene from the partner, and they could assess if the guy got HIV from his partner or not. Is everything clear so far? Or if anyone has a question, we can go back.
No questions so far here in the chat, but please, everyone, don't feel shy and open your mics or type in the chat if you want to.
So let's go to statistical. Oh. So for sample size calculation, they use an effect size of 39% of reduction in the incidence of HIV infection. You might ask yourself why 39%. This happens to be the standard on public health studies when the goal is to assess a preventive measure. So if you see some of the clinical trials from last year for COVID vaccine, some of them use this 39% cutoff. And using this 39% cutoff, they assume that they needed 1,750 serodiscordant couples. So you can see they calculated the sample size, and they used the 39% cutoff. And for data analysis, they used a survival analysis. So in the course, we already discussed about survival analysis. We have discussed a lot of Kaplan-Meier as an alternative to create a survival function. And with Kaplan-Meier, we can estimate the cumulative incidence rate. But they use as well the Cox regression to measure hazard. Before we jump into the results and discuss more about the trial, hazard is sometimes hard to understand. So I wanted to say that hazard is one type of measure of association. When you have measure of associations, you have cumulative instance, and you have rates as well. And hazard is one type of rate, but is like a instantaneous measure of rate. So is also called as mortality force. It estimates it estimates at every point the hazard that the person have. So they use the Kaplan-Meier method, and they use the Cox regression as well. So let's jump to the results. Let's first see about enrollment. As you can see, they enrolled 1,763 couples. 886 couples received the early treatment, and 877 received the delayed treatment. If you see here, there is a depletion of the number of participants during the time. This happens because the study was censored, but it was administratively censored. So when they stop collecting data for the first article, only 43 had gone in the trial for 42 months. All right. In table 1, we can see that the groups were very similar, one from another. Here is about the status of the HIV-1 positive participant and his partner. So you can see that it was a good in terms of baseline characteristics. All right. And let's see some of the results. So something really positive about early treatment is that it was really effective on prevention of HIV transmission. In the early treatment group, only one participant got a linked transmission, and on the delayed treatment, 27. And we are going, when we discuss the limitations of the trial, please pay attention about this one, because we are going to discuss it also. There was also a measure for any transmission. So in this case, it means that the person got HIV from someone else. So in the early treatment, 4 got, and in the late treatment, 35. And here is in terms of the secondary outcomes. So if the early treatment was beneficial for the HIV positive participant, you can say that it was positive. It wasn't so positive as the researchers expected. Here is the Kaplan-Meier curve comparing the transmission between the early treatment group and the delayed treatment. If you see in the bottom, you see that because the study was censored, only few participants contribute for the information at the end of the study. And in the beginning of the study, there was a more expressive contribution. So here it's for linked transmission. Here is for any transmission. And here is for clinical events. So if anyone has a question, please jump in, or we can go back to the shorts as well. So let's first discuss about potential bias that this study had. So when you think bias in a study, it comes in 3 flavors. So you might have selection bias, measurement bias, and confounding as well. One easy way to understand selection bias is with tags. So selection bias happens when the treatment and the outcome causes to the participant to be in the study. Because this was a randomized clinical trial, we don't expect selection bias in the beginning of the study, because if you had a proper randomization, you have characteristics that are similar. But it happens, but you can still have selection bias on the backdoor pathway. This means the treatment affects participants to stay in the study. So if the treatment affects participants to stay in the study, you might still have selection bias. In terms of measurement bias can occur when we don't have a proper measure of exposure and a proper measure of outcome, or even both. And one example of this measurement bias in the study is that one case that I discussed early. So when this article was published, this one case was reported as if the treatment was not effective. In the following studies, they actually compared the virus, and they realized that this transmission happened in the early months of the treatment of the partner. So the partner didn't have any type of virus suppression when this transmission happened. And confounding happens when you have a variable that causes the treatment and causes the outcome. So one example of confounding that could exist in this trial was using condoms. So the participant might be on the treatment group. So he decides, oh, I am covered. I'm not gonna use condoms anymore. And this causes him to infect his partner. And there are unmeasured confoundings creating this pathway. So here with the graph, you can easily understand the difference between confounding and selection bias. And here in the deck as well, here in the top bottom, you can see what is effect modification. An effect modifier is something that changes the relationship between treatment and outcome, but is not related to the treatment itself. So you have different, different stratas in the data. So as we discussed, possible bias in the study was selection bias in terms of participant dropping out. And to see if this hypothesis sustain, we need to see the dropout rate. And the dropout rate was similar between groups. In the treatment group was around 90%, and in the control group was around 86. But you always can do a sensitive analysis in terms of measurement. You can do a sensitive analysis trying to estimate if it was present or not. So the conclusion of the article was that early treatment was positive. It was a really good, really good public health. Sorry, it was a really effective public health. I forgot the word. It was a really good public health strategy, and it changed how society saw prevention of HIV. So after that, many countries changed the drug regimen and started to offer early treatment. And the most accepted initiative today is to a country have a combined prevention with early treatment, offering tests with a regular basis, and other innovations that came after the trial. So this trial was a really breakthrough. It was a really breakthrough. There was observational data before the trial suggesting that early treatment was positive in terms of prevention, but it consolidated this and changed the practice in public health, and changed and hopefully will change how the HIV epidemics happen in the future years. So we can jump to questions and discuss other parts of the article as well.
So Alvairi raised his hand.
Yeah, hi. That was a very nice presentation, Lucas. It's actually very interesting for me because I'm an infectious disease physician, so it's kind of my territory. I really liked hearing about this study that like made it more possible for people to adopt the early treatment. However, may I ask, maybe I missed it, but did you mention anything about how they analyze the data? Did they, how did they like compensate or make up for the missing data from the like large dropout percentage? And one more thing, if I may, was it the analysis like intention to treat? Maybe I missed those points, but if you can kindly answer my questions.
Sure. So in terms of dropouts, let's go to the graph just a second. Oh, no, let's go back. All right. So in terms of, it happens that most of the, it wasn't actually dropouts, it was more related to censoring, because the study was, so there was a board, and the board decided that they had information already. So they stopped when a few participants had contributed for 42%. The trial still happened, and in 2016 they published the result for the entire trial, and it was also around 90% of, so only 10% dropout rate. So this wasn't a big concern for the study, and the data analysis happened as intention to treat. You are right. Something that I forgot to mention actually is that about the treatment itself, this was quite complicated, because when they proposed the trial, they didn't have information what treatment to use. So they partnered with several pharmaceutical companies, and they used a very big list of treatments. So not every participant received the same drug regimen as the others. This, you might see this as a potential measurement bias of exposure, because you're not given the same treatment for everybody. Did I answer your question?
Yes, very much. Thank you. I really appreciate it.
Hi, Lucas.
Hi.
What strategy they use it to assess the missing data? They use it. They established that the data was missing at random, completed at random, or not at random.
So I need to check this in the article. I'm almost certain that they use miss it at random, but I don't want to be wrong. Just a second. It's loading. So I can check and give you your answer later. Okay.
That's fine. Okay.
Let me open here. just a second. If anyone has another question, I can try to answer in the meantime while it loads. Oh, great. So here it has the information here. So they didn't have missing data as missing data, but they had sensory. So they didn't have any missing data for this analysis in 2011.
Okay, thank you.
Great.
So Lucas, Joao Pedro also asked if you can explain the effect modifier concept again.
I didn't hear Alma, so.
Joao is asking if you can explain the effect modifier concept again.
Oh, sure. So here we have a DAG. A DAG is one way to represent a causal pathway. You see that you have the treatment and you have your exposure. You call something as confounding when confounding causes treatment and it causes the outcome. So confounding, we are okay, right? Effect modifier is not a confounding, it's not an error in the study, but it is a difference that exists in real life. So for instance, you might have a different data about the HIV infection depending on gender. But this is not a confounder. It's only because the relationship between treatment and the outcome happens differently between these stratas. So it's not a bias. It's just something that you see different. And when you go to the results, you see that they indeed did in the Cox regression, they adjusted for effect modifiers. So let's go here. Oh, great. So you can see here that being a male, this is not a good example because it's not significant, but let's see here. None of the use were, let's see in the without adjusting. Okay, so you see here that baseline viral load affects the relationship between treatment and the outcome. So depending on the baseline viral load, participant acted differently. So it's something that you have information that is different between strata, but it's not a bias. It's just how the word is. could I explain, or I can try to give a different example? One example would be, so one possible example would be you have the relationship between eating candy and having diabetes, and you know that a certain phenotype it's more prone to develop diabetes, but and when you do your data analysis, you see that someone that has this certain phenotype develop more diabetes. But this is not, you see a strata difference, but it's not like a bias in the study. It's just a difference between the strata. So let's questions. So what do you think about the study design? Did you enjoy it? So what do you think about enrolling couples instead of individuals? Do you think that this was smart or not? What are the pitfalls of doing that?
I think that this was the differential of the study because they used the couple that didn't have the HIV virus viral as the outcome. I mean, they measure if the couple had the transmission after the early antiviral therapy or not. I think that this was a smart decision.
Yes, this is true. Doing the study between couples was smart in terms of measuring infections. But if you think in real life scenario, is not everyone that have HIV that is in a monogamous relationship. So one critic
Sure.
that you might think is that this study don't represent a real life scenario because in real life, to say that it was a public health measure, you need to imagine that everybody might not be in a couple.
Sure.
Yes, and another thing is that
This could actually this could introduce bias, right, to the study in real life situations.
so it affects the external validity of the study because you can't say that it's going to be as effective in real life. But something very nice they did as a clinical trial, if every couple went to visits monthly, and they were, and in these visits, they were actually, so they were taught about how to prevent HIV infection. So they had classes about using condom, how to have safe sex, even if they were not in the early treatment group. So everybody got this information. So it tried to emulate how a real couple would act. And after the trial was published, they offered for everybody to receive treatment, and they followed everybody for more 5 years. And in 2016, they published another article. In the 2016 publication, they had that early treatment was more effective in terms of prevention. They analyzed this one single case that occurred. And because they did this genetic test, they discovered that this transmission happened before the viral load. And after the 5-year period, it came the notion that being undetectable, it's equal to untransmissible for HIV. And this had also a great impact in public health as well. All right.
So Olga raised her hand. Olga, would you like to ask a question live?
No, just to mention that this type of design, and maybe it has to do with a well-coordinated effort. For example, in an effort to coordinate more well the results and all the potential results, testing couples is a more well-coordinated approach. What do you think, Lucas? As you mentioned, what you covered already. I think on that, just an add to add.
So using couples using couples allowed to measure transmission because you were able to test one person regularly, and if you didn't use couple, would be almost impossible to do the study, because you imagine you randomize HIV positive people, and then you ask the person to invite every sexual partner in the last 3 months to go to the center and test for HIV. It wouldn't be feasible to perform a study like that. So this was one way they did in terms of trying to prove that there was not a linked transmission.
Okay, thank you very much. Yes. Mm-hmm.
I think Olga raised her hand as well, right?
Yes, I've already asked the question. I'm all set.
Oh, great.
Thank you.
So yes, the website is called dagti.net. It's really good to use DAGs when you are when you are analyzing your data, and it allows you to understand better the causal pathway. Something very nice about the site, this website, is that is that you create your causal pathway here. Oh, it's hard to, it seemed easier to use than actually is. So you put in DAGT everything you know. So you put your exposure, you put your outcome, and in the website you can adjust or unadjust things. So you create your causal pathway, and in the bottom right of the website you can see the minimal adjustment that you need to do in this trial in terms of to minimize confounding. So it's one way to analyze your data, taking consideration your previous knowledge on the causal pathway. So in the website, you would see in the top left the options for adjusting, and the top right you would see what you should adjust and what you should not. And the website changes the colors depending on that. So you can see here that it's pink. It means that this is creating, this is affecting the true relationship between treatment and outcome. And when you adjust or unadjust things, you see the color change. So it's a very useful tool. Is there any other question? Going back to the background, something that it's really nice, not nice, it's not the proper word, but something that brings a lot of attention is the time it took for HIV to be discovered since it was introduced in humans. So you see there's a big gap between 1908 and 1980s. It took a lot of time to the disease to get knowledge. And this is more related how society is organized. With development of cities in Africa, the environment for creating new zoonoses became more fertile. So it became easier for a disease to spread. So it took almost, if you see here, it took almost 40 or 50 years for the virus go from Kasasha region to East Africa. And if you imagine COVID today, it took only 2 months to spread the entire world. Of course, that COVID, you have the symptoms when you are infected, and in the case of HIV, it takes time, almost 10 years to develop a clinical outcome. But it's still, you see how so you can see how it changed over the time. And you can see also the great impact you can see the great impact that if mankind, when mankind explore the edge between cities and non-habited areas, and how this might have a public health effect in the future in terms of introduction of new zoonoses. So we need to be careful if you think about expanding the rural areas and expanding cities all over the world with deforestation.
Any other questions, everyone, before we end up the section?
Hi, Lucas. This is not a question, actually. I just wanted to say that it's really nice that you chose this trial, because I really appreciate the fact that you gave it a sense of comparison with the pandemics that we have today. And as you said, it's a great take-home message that we should be careful as the point that we have as humankind in the current situations that we're dealing with and what we had in the past. So it's great that you presented this trial today, so we can what we have already seen and like implement it to the future. Thank you for your presentation today. It was great. You did a great job.
Thank you. Ali, right?
Yeah.
Thank you. Oh, it's because I don't see the zone. Thank you, Ali. And thank you, everybody, for joining today. If you are shy and you don't want to ask the question aloud, you always can send me an email and we can discuss the trial later.
Okay, thank you everyone for joining today, and thank you, Lucas, for preparing this great session for the students and colleagues in PPCR. We really appreciate your time and your dedication. Have a nice night, everyone, and we will share the recording and the slides later. Thank you.
Thank you, Alma, and thank you, everyone. It was my pleasure. I hope I see you later, everybody.